Assalamu alaikum dear aimers, hope so you all are doing well. Today the topic of our discussion is cyclic photophosphorylation. In the last video we had a detailed discussion about forms of light reactions, then phosphorylation and then non-cyclic photophosphorylation. And today we'll discuss cyclic photophosphorylation. Now what are the contents for today's topic? We will look at the cyclic photophosphorylation, then its definition, its properties and whole the process. Okay, let's start the topic. Now, we had a detailed discussion about phosphorylation, that addition of phosphate to an organic compound is called as phosphorylation. For example, addition of phosphate to ADP to make ATP is phosphorylation. And photo means light. As the production of ATP takes place in the presence of light, so it is called as photophosphorylation. And the flow of electrons throughout the process is in cyclic manner. That's why it is called as cyclic photophosphorylation. So what is the definition of cyclic photophosphorylation? The production of ATP means phosphorylation by light dependent reactions means photo in the presence of light in a cyclic electron flow in a cyclic pattern is called as cyclic photophosphorylation. So cyclic photophosphorylation is basically a form of light reactions. Okay. Now, where does cyclic photophosphorylation take place? Cyclic photophosphorylation, like that of non-cyclic, takes place in thylakoid membrane. And what are some properties associated with cyclic photophosphorylation? First property is that it is a non-continuous process. It does not occur continuously in a plant cell. It only occurs sometimes in a plant cell. The second point is it is not a normal routine process. Like non-cyclic photophosphorylation was a normal routine process. But cyclic is not a normal process. It just occurs under specific conditions in the plant cell. And third is it's just a temporary process, not a permanent process. There is some time that the electron flow shifts from non-cyclic to cyclic under certain conditions. Now we will look at what are that certain conditions under which a plant cell or a chloroplast shifts from non-cyclic pattern to the cyclic pattern of electrons. Now there are two conditions for the cyclic photophosphorylation. First condition is the low level of ATP in chloroplast. Sometimes in non-cyclic photophosphorylation, there is not so much production of ATP. Like the level of ATP is not that much which is required for the Kelvin cycle. As you know that the energy absorbed by the electrons is transferred to the ATP is converted into the chemical energy of ATP, and this chemical energy of ATP is then transferred to glucose in Kelvin cycle. So sometimes the level of ATP production is not that way which is required for the Kelvin cycle. That would be the time when non-cyclic photophosphorylation will be stopped and cyclic photophosphorylation will start to be continued. Now, when does the level of ATP falls? First of all, you saw in the non-cyclic photophosphorylation what was happening, that the ATP was synthesized on the electron transport chain of photosystem II, and then on the electron transport chain of photosystem I, NADPH was synthesized. So what happens that the level of ATP falls and NADPH accumulation continues because ATP is not produced the way it should be produced, but NADPH is produced as usual. So ATP level decreases and NADPH level increases in the chloroplast. That's why NADPH accumulation occurs. And when the accumulation of NADPH occurs, this accumulation is basically a stimulus to shift the electron flow from non-cyclic to the cyclic phosphorylation. Got it? So these two, low level of ATP and accumulation of NADPH, these both are the conditions that would start the cyclic photophosphorylation. Now, what are some important points that you have to remember for the cyclic photophosphorylation? First point is the reactants. Only light energy is the reactant of cyclic photophosphorylation, while water was also the reactant in the non-cyclic flow. But only light energy is the reactant in the cyclic photophosphorylation. Now, what are the products? Only ATP is produced in cyclic photophosphorylation, while in case of non-cyclic, NADPH was also produced. But in case of cyclic, there is no production of NADPH, only ATP is produced because plant cell or a chloroplast requires ATP. Plant cell is running low on ATP. So ATP is produced in case of cyclic photophosphorylation. Now, what is the flow pattern of electrons? This is very obvious that electrons flow in a cyclic pattern. The electron donor and electron acceptor, they both will be the same and then they will form a cycle. That's why it is called as cyclic photophosphorylation. Now, what the type of photosynthesis it is? This is basically an anoxygenic photosynthesis. Anoxygenic photosynthesis means the type of photosynthesis in which oxygen is not released. So it is without oxygen, so it occurs both in aerobic and anaerobic conditions. Now, how many photosystems are involved in cyclic photophosphorylation? Only photosystem 1 is involved in cyclic photophosphorylation. So do remember all these points, and you can also write the points in your exam so your paper will be more effective. Now, what are the steps involved in the cyclic photophosphorylation? There are 2 steps involved in the cyclic photophosphorylation, and do remember how many were there in non-cyclic? Yeah, there were 6 steps in the non-cyclic photophosphorylation, while in case of cyclic, there are only 2 steps. First was light absorption by photosystem 1, because only photosystem 1 is involved. And second step is ATP synthesis, because the main purpose of cyclic photophosphorylation is to increase the level of ATP in chloroplast. So students, first step is the light absorption by photosystem 1. The process is entirely same as that of the non-cyclic photophosphorylation. What happens that the light energy falls on the chlorophyll A molecule of reaction center of photosystem 1, which can absorb maximum wavelength of 700 nanometer. Antenna pigment molecules trap all of the light energy and transfer energy towards the chlorophyll A of reaction center, and by absorbing light energy, the electron is excited to higher energy level. Now do remember this point that in the non-cyclic photophosphorylation, we saw that 2 electrons were excited to a higher energy level, because 2 electrons were required to reduce NADP plus into NADPH. If you haven't seen that video, then go and check out that video first, and then you will have a better understanding of cyclic photophosphorylation. So in that case, 2 electrons were excited, but now only 1 electron is excited because there is no production of NADPH. So we don't require 2 electrons to reduce NADP plus into NADPH. So only 1 electron is excited. Now the electron is excited from chlorophyll A, an electron hole is created, the deficiency of electron or a positive charge is created on photosystem 1, and that hole, that deficiency of electron and that positive charge needs to be filled, and that is filled by the photosystem 1, because it's a cyclic pattern. Electrons will be excited from photosystem 1 and will be returned to the photosystem 1. So the electron hole will be filled by the photosystem 1, and then the excited electron is trapped by the primary electron acceptor. You know all these points very well if you have taken the previous lecture. Now, how is ATP synthesized in the cyclic photophosphorylation? We know that only photosystem 1 is involved. You see here that these lighter green molecules are antenna pigment molecules, chlorophyll A, chlorophyll B and carotenes. Light energy fall on these antenna pigment molecules and channelized towards the reaction center chlorophyll A. Now, from chlorophyll A, electrons absorb light energy, and then the electrons are excited to higher energy level. You see that it's a higher energy level and electrons are excited from it. And these electrons are then trapped by the primary electron acceptor of photosystem 1. Now, from primary electron acceptor, these electrons travel to the new substance, which is ferredoxin. In the last video, we saw that ferredoxin was involved in the electron transport chain. So it's an electron carrier, iron-containing electron carrier. So electrons are channelized towards the ferredoxin. But now the electrons will not move to NADP reductase, because already there is accumulation of NADPH in the cell. So there is no need to synthesize NADPH. There is deficiency of ATP, so ATP needs to be synthesized. So electrons don't go down the chain and to NADP reductase. Rather, from ferredoxin, electrons are transported to the cytochrome complex. Because you all know, we saw in non-cyclic photophosphorylation that whenever electrons go from cytochrome complex to plastocyanin, then the energy, there is a difference of energy level, because you know that the energy level goes on decreasing. Okay, so electrons release energy, and that energy is used to synthesize ATP. So similarly, electrons from ferredoxin alter their path and go to cytochrome complex, and from cytochrome complex these electrons travel to the plastocyanin. As you know that this is high energy level, the electrons have a lot of energy here, while traveling down, this is the low energy level. So whenever electrons travel from high energy level to low energy level, they release all of the energy that was absorbed by the electrons in the form of photons. So electrons will release all the energy that they absorbed from the photon, and that energy is used to combine ADP with the phosphate to make ATP. Now all the light energy which was absorbed by the electrons is now transferred to ATP in the form of chemical energy stored in the phosphate bonds of ATP. This ATP is extremely important for the Kelvin cycle because this ATP, the energy stored in this ATP, will be used to combine carbon dioxide and hydrogen to make triose in the Kelvin cycle. And then electrons, by releasing all of the energy, come back to the photosystem I to fill the electron hole which was created. Okay, electrons got excited from photosystem I, then went to primary electron acceptor, then to ferredoxin, and then to cytochrome complex. And as electrons travel from cytochrome complex to plastocyanin, it is low energy level, so all the energy is released and is used to synthesize ATP. Now electrons travel back to the photosystem I to fill the electron hole which was created by the excitation of electrons. So you see, it's a cyclic pattern. So that's why it is called as cyclic phosphorylation. And phosphorylation means synthesis of ATP. As the whole process is taking place in the presence of light, so it is called as photophosphorylation. Photo means light. Okay, now ATP has been synthesized, but there is no NADPH production, and only photosystem I is involved in the process. Now I made this diagram only for your understanding, but you will make the diagram which has been mentioned in your book. So this is the diagram which has been mentioned in your book. In this diagram you can see that this is the colored part. It means that only these structures are involved in cyclic photophosphorylation. So you see here, this is also a cyclic path. You see that electrons are excited from photosystem I, they go to primary electron acceptor, and then to ferredoxin, then cytochrome complex, and then again to plastocyanin. The energy of electron is used to make ATP. I told you in the last video that synthesis of ATP is called as phosphorylation, while the mechanism by which ATP is synthesized is called as chemiosmosis, which will be our next topic of discussion. So you see here, all the colored parts are involved in the process, while all the non-shaded areas, these black areas, are not involved in cyclic photophosphorylation. So what are the structures which are not involved in cyclic photophosphorylation? Photosystem II, primary electron acceptor of photosystem II, plastoquinone. Do remember that this plastoquinone is not involved. Most of the students, what do? That they transfer the electrons from ferredoxin to plastoquinone. No, electrons are transferred from ferredoxin to cytochrome complex, because cytochrome complex is basically involved in the synthesis of ATP, which we will discuss in the chemiosmosis. Okay, so electrons are transferred from ferredoxin to cytochrome complex and then to plastocyanin. Okay, this NADP reductase is not involved because there will be no NADPH synthesis, and NADPH is also not produced in cyclic photophosphorylation. Now I am going to compare both cyclic and non-cyclic photophosphorylation. So here is the comparison chart of both of the phosphorylations. Firstly, look at the non-cyclic photophosphorylation. First point is its definition. I mentioned it in the lecture. The production of ATP by light-dependent reactions in non-cyclic electron flow is non-cyclic photophosphorylation, while production of ATP in a cyclic electron flow is cyclic photophosphorylation. Now, initial donor and final acceptor of electrons are different for non-cyclic. Electron is donated by photosystem II and then by water, and the final acceptor of electron is NADP reductase to make NADPH. While the initial donor and the final acceptor of electrons are same in cyclic photophosphorylation, because electrons are donated by photosystem I and electrons are also accepted by photosystem I. That's why it makes a cyclic pattern. Now the third difference is that photosystem I and II both are involved in non-cyclic photophosphorylation, while only photosystem I is involved in cyclic photophosphorylation. In non-cyclic photophosphorylation, photolysis occurs, while in case of cyclic photophosphorylation, there is no use of water, so there is no oxygen production, so photolysis does not occur. In case of non-cyclic, oxygen is produced because water is used. In this case, oxygen is not produced because water is not used. Light energy and water are the reactants. Okay, both of these components are the reactants, while in case of cyclic, only light energy is the reactant, because water fills the electron hole in photosystem II. As photosystem II is not involved in this process, so there is no need of water, there will be no release of oxygen, and there will be no photolysis. The next point is that ATP and NADPH are the products. Both of these products are formed, while in case of cyclic photophosphorylation, only ATP is the product because chloroplast was running low on ATP. And the last point is that non-cyclic photophosphorylation is a normal routine process in plants. That in a normal routine or on daily basis, non-cyclic photophosphorylation takes place in plants. But if ATP is low in the chloroplast, then it's a temporary circuit. It's just a short circuit. You saw that I mentioned in the previous slide that cyclic photophosphorylation is a short circuit, which means that only photosystem 1 was involved. The pathway was short. It was not Z scheme. So pathway was short. That's why it's called a short circuit. While cyclic photophosphorylation, it's just a temporary process. Whenever the demand of ATP is fulfilled, the cycle will immediately shift to the non-cyclic pattern. Okay, so normally non-cyclic photophosphorylation occurs, but when ATP is low, then cycle shifts to cyclic photophosphorylation. And this is the difference between cyclic and non-cyclic photophosphorylation. This is the very important short question, and sometimes it can also come as a long question too. So if you will write just write 3 to 4 points in short question, but write all the points in a long question with headings, then it would be very effective. Now students, here is the summary of the lecture. We saw that only photosystem 1 was involved in cyclic photophosphorylation. Only 1 photon was used, while 2 photons were used in case of non-cyclic. Only 1 electron was excited because there was no need for the reduction of NADP+. That's why only 1 electron is excited, and the product was ATP. No NADPH, no oxygen, only ATP. Okay, so students, this is your processing time. This is your time for some task. And what are your tasks to do? That firstly, you have to practice the diagram of cyclic photophosphorylation at least 2 times right now sitting here. Okay, promise me that you will practice this diagram. The diagram is exactly same to that of non-cyclic. What you only have to do, that you have to make the structures involved in cyclic photophosphorylation more shaded, or give them any other color, or make them with the help of marker, and the structures which are not involved, then make them with the help of pen. Okay, so make some difference between both of them. Okay, so do practice it at least 2 times, and then attempt this question. Take it as long question. Okay, that write the difference between cyclic and non-cyclic photophosphorylation sitting here, learn the differences which I mentioned in the slide. You can take screenshot and then write the difference between photosystem 1 and 2. I told you the difference between photosystem 1 and 2 when I was teaching you light reactions. Okay, so you may go and check out that topic. So you have to write that differences on your notebook now. Okay, now means now. Students, if you have got the lecture, have a big smile on your face. Be happy, make others happy. Don't forget to like, share and subscribe to my YouTube channel. See you in the next video. Allah Hafiz.